Well, good morning, good afternoon, or good evening, ladies and gentlemen. Thank you for joining us for our second installment of our video PowerPoint series for Unit 5.5, Mitosis and Meiosis. Today, we're going to be talking about meiosis. Now, once again, I'm sorry, but this is only going to be an audio version of our video. We'll still go through all the animations, same as last time, but there will not be any thumbnail of a video in the upper right-hand corner. Just my voice today, so to make up for that, here's Macho Man Randy Savage. Once again, take it all in. Alright, so today what we're going to be talking about is meiosis. So meiosis is primarily used for replication of sex cells, like the sperm cell in the male and the egg cell in the female. So the first question we have to ask ourselves is, what is an N? So yes, I'm sure you're all aware that N is a letter, but in biology, N has a bit of a different meaning. N represents what we call the haploid condition. So haploid, we're talking about half the genetic material, or the number of chromosomes in a gamete. So the number of chromosomes found in a gamete, in humans at least, is 23. So remember, the haploid condition for human cells is 23 chromosomes. Diploid, or 2N, is the number of chromosomes found in a somatic or body cell. We get one copy from the mother, one copy from the father. And what these are homologous pairs of chromosomes. So we have homologous, meaning we have two chromosomes for the same trait. So things like eye color, hair color, etc. Now those are a bit more complex, but I'm just using that as an example. So let's continue. We have two types of diploid cells in sexual organisms. We have the somatic cells and the germ cells. Somatic cells are body cells, which are the diploid condition. They divide by mitosis to make exact copies, and they're found in every tissue except for reproductive tissue. In the meantime, germ cells, which is short for germinating or reproductive cells, not like bacteria germs, these are also diploid, but most divide by meiosis to make gametes, or sex cells, so the sperm in males and the ova or eggs in females. These can be found in the testes in males and the ovaries in the females. So mitosis versus meiosis. So we have a quick little chart here. Mitosis is asexual reproduction, whereas meiosis is sexual. In mitosis, we create body cells, or somatic cells. In meiosis, we create sex cells, or gametes. In mitosis, the DNA replicates once. Likewise in meiosis. In mitosis, we only have one set of divisions, where in meiosis we have two. At the end of mitosis, we have two identical daughter cells, whereas in meiosis we have four different daughter cells, each one containing a unique set of genetic information. In mitosis, they create diploid cells, which have the condition 2N. Whereas in meiosis, we create haploid cells which have the condition N, like we discussed a couple of slides back. Meiosis is part of sexual reproduction, requiring two parents, much like Happy Feet down here. So in meiosis, one diploid germ cell divides to become four haploid gamete cells. So in order to go from diploid to haploid, meiosis involves two divisions. We have meiosis I and meiosis II. During meiosis I, we split the homologous pairs of chromosomes, whereas in meiosis II, we split sister chromatids apart, which we'll go through shortly. So it's time to go ahead and name the phases. Now if you know mitosis, you know meiosis. All we do is just double what we do. So we have prophase 1, metaphase 1, anaphase 1, and telophase 1. After that, we have cytokinesis. For the second meiotic division, we have prophase 2, metaphase 2, anaphase 2, and we finish it up with telophase 2 and cytokinesis once more. So once again, you don't have to make these tables. If you go ahead and look in your workbook, I'm sure you'll find them. So as we go through, let's go ahead and work through prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. So we'll go through the first division, what it looks like, and what's happening. And once we take care of that, we'll move into prophase, metaphase, anaphase, telophase 2, along with the second round of cytokinesis. Now, like I said before, you don't have to go ahead and draw all this. This can be found inside your workbook. So, I'll see you on the next couple of slides, and we'll start to fill this thing in. So during prophase 1, once again the nuclear envelope disintegrates. Our centrioles migrate and produce spindle fibers. But now, our homologous chromosomes pair up. So homologous chromosomes, we're talking about things like unique individual traits, like eye colors, hair color, dimples, things like that. And what happens here are the homologous pairs cross over. So as you can see, we don't have one copy now. We have two copies of our homologous chromosomes. We have a copy here, and we have a copy here. Now what they do is go through something called crossing over. The crossing over we'll talk about on the next slide. So crossing over is an exchange of segments between genes between homologous chromosomes. So let's say this is for hair color, all right? Let's say blue is for, you know what, let's just get ridiculous. Let's say that's for blue hair. Why not? And let's say this X over here is for red hair. So what we have primarily is, since they're both genes for hair color, they can actually swap parts of themselves over. So as you can see, down here, we have part of the red mixing with part of the blue, and part of the blue mixing with part of the red. So they can actually exchange parts to go ahead and create more variants. Now the more variants we have, the better, which we'll talk about more when we get to genetics. But all you need to know for now is crossing over is exchanging two parts of the same type of chromosome to create more variation. So, metaphase 1. During metaphase 1, homologous pairs of chromosomes line up on what we call the metaphase plate. So both pairs of chromosomes line up on the center, just like we did with mitosis. During anaphase 1, the homologous pairs are pulled apart to opposite poles. So we go ahead and separate them. Then we finally hit telophase 1 and cytokinesis. Here, the chromosomes are no longer homologous pairs, because as you see, now we have one on this side and one on this side. They're no longer paired up with each other. Once this happens, the chromosomes then prepare for what we call meiosis II. And the cytoplasm will then begin to split into the first two of 4 daughter cells. So, after we go ahead and go through telophase, we'll start to go through cytokinesis and split the cells up into 2. Note, so please take note of this. At the end of meiosis I, daughter cells are already haploid. So they already have half the information that they started off with, because homologous pairs were already separated. Now, the 2 haploid daughter cells must then go through meiosis II. In meiosis II, the 2 haploid cells will separate sister chromatids to make 4 gametes. So basically what we're doing is we're going through mitosis with each cell individually. So during prophase 2, the centrioles migrate and produce spindle fibers, just like in mitosis. During metaphase 2, the chromosomes line up along the metaphase plate, just like in mitosis. Noticing a pattern? I think so. Anaphase 2, sister chromatids are pulled apart to the opposite poles, just like in mitosis. Wonderful. Finally, we hit telophase 2 and cytokinesis. So the nuclear envelope reforms around the 4 daughter cells, and the cytoplasm splits to create the 4 separate cells. Each of the 4 daughter cells is a new gamete with genetic information different from either parent. So as you can see here, each of the 4 daughter cells has a different combination. So, since they each have a different combination, that means that whatever one actually goes ahead and fertilizes the egg or is fertilized by the sperm is going to contain slight variations of the original parent. So, based off what we have here, is this meiosis I or meiosis II? I'll give you a little bit of time to think. Okay, if you said meiosis I, you'd be correct. The reason behind this is because we only have one set of divisions. So at the end of this, we have 2 daughter cells instead of 4. Now, based off that, what we have here is without a doubt meiosis II, because we're going from 2 cells to 4. So if you think back, when do these cells become haploid? If you said at the end of meiosis I, you'd be correct. Why? Because what we did here is we separated the 2 homologous chromosomes, making them haploid. So here's a nice breakdown of what we just talked about. During mitosis, we start off with, in humans, 46 chromosomes, or 2N. Now, we're just making identical copies of the original cell, right? So during the DNA replication, we go from 46 chromosomes to, if we double that, 92 chromosomes. So when it splits, each cell has 46, making it 2N. Now with meiosis, the same thing happens initially. We go from 46 to 92 to 46, eventually to our haploids, which now each have 23. So, in sex cells, this is how it looks. Did you know that there's 200 to 400 million sperm per ejaculate? Fun fact. Now, here's the thing though, what happens to the 3 other eggs? Now, normally only 1 egg can get fertilized at a time. So what normally happens is whichever is the largest of the eggs will actually absorb the others. So usually only 1 wins per cycle. So finally we reach fertilization. This is when the male and the female gametes unite, so the sperm meets with the egg. The fusion of 2 haploid nuclei produce a diploid nucleus, which makes sense because if you think about it, what we're going ahead and doing here is we're taking 23 from dad, 23 from mom, and when they come together, we get our first body cell that has 46 chromosomes. What we call this is the zygote. So it's when the egg and the sperm meet and combine. So the zygote is the new life in one diploid cell. So meiosis and sexual reproduction guarantee variation and diversity in 3 specific ways. Number 1 is independent assortment. This is where the homologous chromosomes separate randomly, so you never know which way they're going to go. You don't know if one combo is going to be pulled to the left, one combo to the right. That's just it. It's like a box of chocolates. You never know quite what you're going to get. Number 2 is crossing over. So the homologous pairs exchange gene segments. This, once again, like we said earlier, crossing over creates more variation. And the more variation, the better. And finally, we have random fertilization. So like we said before, with the sperm, there's 200 to 400 million variations that could fertilize that egg. So as you can see, the sheer number of possibilities is astronomical. So which sperm meets which egg? It's all a game of chance. Well, ladies and gents, that just about goes ahead and closes up another lesson here for our video PowerPoint series. I'd like to thank you all for going ahead and joining us today as we talked about mitosis and meiosis. Well, today we talked about meiosis, we talked about mitosis last time, so if I didn't thank you back then, I thank you now. So today we talked about why meiosis is important, how the variations are made, and the steps. So, until next time, I will see you in the next video. Until then, you all keep it classy.